Welcome back, biologists, to your next biology screencast. In this screencast, we'll begin Chapter 3 and talk about cell structure and function. Cells are the basic building blocks of life, and this diagram shows many of the different types of cells found in the human body. This chapter's case study focuses on penicillin, which was considered a wonder drug in the 20th century. Penicillin was discovered by a biologist named Alexander Fleming. In 1928, he observed that a fungus called Penicillium notatum was capable of killing many kinds of bacteria. This infographic from the book shows what led to Alexander Fleming's discovery of penicillin. The fungus, specifically a mold, inhibited the growth of bacteria around it on a petri dish. Let's talk about what an antibiotic is. Antibiotics are chemicals that can slow down or stop the growth of bacteria. They're often produced by living organisms. For example, in our case study, the antibiotic penicillin is produced by a fungus, a mold called Penicillium notatum. Antibiotics preferentially kill bacteria without harming the human or animal host. They do this by targeting what's unique about bacterial cells. They take advantage of the differences between bacterial and animal cells. The cell theory describes how we understand cells. It states that all living things, also called organisms, are made of cells. It also states that every new cell comes from the division of a pre-existing cell. This infographic shows cells from different organisms. It also reiterates that cells only come from other cells. We'll talk about where the first cells came from later on in the course. All living things are made up of one of two basic types of cells. All bacteria are made up of prokaryotic cells. Prokaryotic cells lack internal membrane-bound organelles, like the nucleus or mitochondria you may have learned about in past science courses. All other living things, including us, are made up of eukaryotic cells. These cells do have internal membrane-bound organelles. The infographic on this slide shows the differences between a basic prokaryotic cell and a basic eukaryotic cell. All cells, whether prokaryotic or eukaryotic, have four characteristics in common. First, they all have a cell membrane made up of phospholipids and proteins. Second, they have cytoplasm, a gelatinous, aqueous interior, aqueous meaning watery. Third, they all have ribosomes. Ribosomes are a complex of RNA and protein that carry out protein synthesis. Basically, ribosomes are protein factories. And lastly, all cells have DNA as their molecule of heredity. Eukaryotic cells differ from prokaryotic cells because they contain a nucleus and other membrane-bound organelles. The nucleus contains the cell's DNA and is basically the cell's control center. Only prokaryotic or bacterial cells contain a circular DNA chromosome floating freely in the cytoplasm. They also have no membrane-bound organelles. And finally, they have a peptidoglycan cell wall. This is a rigid structure surrounding and enclosing the cell membrane. Peptido refers to amino acid elements of the cell wall, and glycan refers to monosaccharide sugar elements of the cell wall. Peptidoglycan is what makes bacterial cells rigid. Peptidoglycan is a polymer made up of sugar and amino acid molecules. The peptidoglycan cell wall allows bacteria to survive in watery environments, like the interior of your body. What penicillin does is weaken the cell wall and make it leaky. As a result, bacterial cells will fill up with water and burst due to a process called osmosis, which we'll talk about later. This infographic reviews how penicillin weakens the bacterial cell wall by interfering with its synthesis. As a result, water rushes in and causes the cell to burst. Osmosis is the movement or diffusion of water across a semi-permeable membrane from an area of lower solute concentration to an area of higher solute concentration. The easiest way to think about this is that water will move from where it's more pure to where it's less pure. Things like salts that are dissolved in the water will affect its purity. Cells can be in one of three different environments or solutions that will affect the movement of water. These environments or solutions are called hypotonic, hypertonic, and isotonic. In a hypotonic solution, water will flow into a cell, causing it to swell and even burst. In a hypotonic solution, solute concentrations are lower and water concentrations are higher outside the cell. In an isotonic environment or solution, water flows equally in both directions, and the cell does not change in size or shape. Isotonic solutions tend to have equal solute and water concentrations both inside the cell and outside the cell. Remember, the prefix iso means same. In a hypertonic environment or solution, water will flow out of the cell, causing the cell to shrivel, or if it's a plant cell, to wilt. Hypertonic solutions tend to have higher solute concentrations and lower water concentrations outside the cell. After penicillin was discovered in 1928, it went into wide-scale production in the 1940s. This happened just in time for World War II. Soldiers were treated with penicillin, and penicillin was considered a wonder drug. Before that, an infection could be just as dangerous as combat. Penicillin doesn't work on all bacteria. That's because we can divide bacterial cells into two different types. The first type is called gram-positive. Gram-positive bacteria have cells that have the peptidoglycan cell wall that will retain a purplish gram stain. This can be seen under a microscope, like in the picture shown on the slide. Bacteria can also be gram-negative. Gram-negative bacteria have a lipid membrane that surrounds the peptidoglycan cell wall and does not retain the purplish gram stain. This lipid membrane prevents penicillin from reaching the peptidoglycan cell wall underneath. The photo on this slide shows both purplish gram-positive bacteria and pinkish gram-negative bacteria. This diagram shows the differences between gram-positive bacteria and gram-negative bacteria. The gram-negative bacteria has an extra outer membrane. Because penicillin doesn't work on all bacteria, we need other antibiotics to treat infection. Streptomycin is another example of an antibiotic, and it works by interfering with prokaryotic ribosomes. It leaves eukaryotic ribosomes alone. The infographic on this slide shows how antibiotics like streptomycin stop the function of bacterial ribosomes but leave human ribosomes alone.